When we talk about carcinogenesis, what are some important things you always have to remember? And that is it's a multi-step process, and the development of all human cancers requires the accumulation of multiple genetic changes. So we always think of neoplasia as a genetic disease. And it can be inherited from germline mutations that are passed on from each generation, or during life or in utero, you could acquire new mutations that increases the likelihood of going on to neoplasia. And when you think of a tumor in general, you have to remember there's over 205 different cells in the body. And each cell can go through mutational changes, give you either a benign tumor or malignant. So when you think of cancer, it is many diseases, hundreds to thousands of diseases, because you can think of all the different permutations that one could see in terms not only between benign and malignant, but also the histologic type as well. And the most important mutations involve what genes that they will ask you about are going to be the proto-oncogenes, and the way you think of proto-oncogenes as growth-promoting genes, like growth factors, receptors that bind to them, the transduction of the signal, the GTP binding protein, or what is called the RAS proto-oncogene, and MYC and so forth. Then the other category you have to remember is growth inhibiting genes, and we call these tumor suppressor genes, and the classic one would be like p53, which is important for preventing mutated cells going through the cell cycle and therefore go through the process of apoptosis. And these genes of tumor suppressor regulate what? Apoptosis, senescence, as well as growth arrest. When we talk about activation of growth-promoting oncogenes or proto-oncogenes, these are, you have to remember, are normal cellular genes in eukaryotic cells. They're important to tell the cell to proliferate as well as to follow the different lineages of differentiation. They, in terms of proto-oncogenes, can be a change in the gene sequence where you can generate a new gene product. It can have the loss of the gene where regulation, where you have overexpression, so you have more number of polypeptides for that gene product. The mechanisms of oncogene activation are the following. You think of point mutations, chromosomal translocations like the Philadelphia chromosome in CML where it's 9 to 22. You think of gene amplification as well as insertional mutagenesis. And these activated oncogenes have now lost the regulatory control either by translocation, amplification, or mutations. Therefore, cellular proliferation is unchecked and leading to the eventual process of neoplasia. Here is a table, one of two, that in this case we're going to look at proto-oncogenes. The other table that we'll discuss in a moment are the tumor suppressor genes. Here in this table you can see on the left is the oncogene and its nomenclature that's used, the type of tumor or tumors that one sees associated with these proto-oncogenes, what is the gene product, and the mechanism of activation that will lead as one of the steps to neoplasia. Remember, there's over 100 proto-oncogenes that are described in the literature. Obviously, you're going to pick the ones on these on this particular type of exam to basically illustrate common tumors, unless there's something unique about these proto-oncogene that is illustrative, that's an important principle to learn, and that's how they're going to construct these type of questions. And that's characteristic of any type of disease process. So starting with growth factors, we have basically HIST1 or INT2, and you'll notice if you look at this list that it's lowercase letters as well as 3 letters that are used in the nomenclature of proto-oncogenes. In this case, HS2-1 and INT2, basically the gene product is fibroblastic growth factor or fibroblast growth factor, and essentially it's associated with stomach, breast, urinary bladder, as well as melanoma. You don't have to memorize all those different tumors. They're not going to ask that, but when you hear fibroblastic growth factor as a gene product, you think of these proto-oncogenes. When we talk about another growth factor, we've talked about this in terms of platelets, platelet-derived growth factor. It's also made by macrophages. And what is the most common primary tumor of the brain are astrocytomas. And so it's SIS or CIS. Probably one of the most important proto-oncogenes that you'll come across that will probably ask you about are the growth factor receptors. And since the majority, or 90% of malignant tumors come from epithelial origin, because you think of epithelial cells like the skin, the stomach, or the GI tract, I should say, the urinary tract, respiratory tract, they're exposed to the environment, whether it's radiation, as well as chemical carcinogens, that the likelihood of acquiring mutations, and since they're proliferating and have stem cells, is greatly enhanced. So when we talk about epidermal growth factor receptor, we think of breast cancer. So the proto-oncogene ERB-B2, we think another name is NU2, or what's used today is HER2. In this case, they capitalized it. And it can be also seen in ovarian and lung cancer, but it's epidermal growth factor and its amplification. In the case of breast cancer, clinically what it's used for is on a lumpectomy or a mastectomy specimen. When they look at the breast cancer, they want to know whether or not HER2 is overexpressed, is because it has a prognostic factor. If HER2 is expressed as a poor prognosis versus non-HER2 expression. If we go to the next set of proto-oncogenes, RET, which is the glial neurotropic factor receptor, and rare diseases that are autosomal dominant is MEN2 and 3, where remember you have the rare tumors of pheochromocytoma and medullary carcinoma of the thyroid. Then a famous translocation of 9 to 22, we think of chronic myeloid leukemia and acute lymphoblastic leukemia, and you get this BCR-ABL fusion protein, which is basically encoding the ability to phosphorylate tyrosine residues. Remember from biochemistry, how do you regulate proteins? An enzymatic process or structural proteins is basically phosphorylating 3 amino acids. The most common one is serine, threonine, and in this case, the ones that are associated with proto-oncogenes is tyrosine. And it's a translocation, again, of 9 to 22. And another important class of proto-oncogene is RAS. Now, RAS is a GT binding protein that's important for the transduction of signal from receptor in the plasma membrane bound by a ligand. And GTP binding protein basically become important in the ability to transduce signal. So if you have a mutation, a point mutation in this case for the RAS, you don't need a growth receptor or a growth factor binding to it. It'll constantly be turned on and the transduction will be essentially unchecked in terms of regulation. So KI, these proto-oncogenes were first studied in retroviruses, and the retroviruses, when they infected mammalian cells, they would pick up these proto-oncogenes, and so the KI refers to Kirsten virus, where it was studied. Now we come to CMYC, and the C means cellular. MYC is a transcription or nuclear regulatory protein, and that translocation, you think of 8 to 14, you think of the case of Burkitt's lymphoma. Now, in the MYC family, you also have LMYC. L is capital for the organ, the lung. And so essentially, we think of small cell lung carcinoma, which is going to be a regulatory protein, and its amplification. NMYC, N stands for the capital letter N for one of the most common solid tumors found in the pediatric population is neuroblastoma. And then we come to cell cycle regulatory proteins. You think of BCL-1 for mantle cell lymphoma. It involves cyclin D protein. Remember the cyclin proteins A through D are important for various stages going through the cell cycle. And then there's CDK4 for melanoma and glioblastoma multiforme, which is the most common primary malignant tumor of the brain. And it's a cyclin-dependent kinase, and it's associated with amplification. In terms of tumor suppressor genes, how do we inactivate this? Well, obviously these tumor suppressor genes are going to encode proteins that are going to regulate and suppress cell proliferation that are initiated in the progression through the cell cycle. And the mechanism of action of tumor suppressor genes, like P53, is to prevent a cell that's damaged. It must be important, I've reiterated it multiple times, from entering the S phase. And then the retinoblastoma gene, RB, prevents the cell from entering the S phase until appropriate growth signals are present. And then, as you recall, in studying carcinogenesis, they have what is called the Knudsen two-hit hypothesis, states that both tumor suppressor genes must be inactivated for oncogenesis. And again, it's a reflection of the multi-step process of carcinogenesis. When we talk about tumor suppressor genes, the mode of action of inherited germline mutations, we think of first hit inherited germline mutation carrying from one generation to the next. And then the second hit is going to be acquired somatic mutation, whether it's from a virus or some type of chemical carcinogen that will alter that gene that will be important for the progression to neoplasia. What are some examples of these inherited germline mutations? You think of familial retinoblastoma involving the RB gene. And basically, the RB gene is found on chromosome 13. And then you can also have a high rate, obviously, of retinoblastoma, but also can involve other organ systems that increase the risk for osteogenic sarcoma of the bone. Lee-Fraumeni syndrome is a rare condition where you have a germline mutation of P53 on chromosome 17, and you have a high rate of many types of tumors associated with it. Because remember, what's probably the most common tumor suppressor gene or cancer gene in the majority of neoplasias is P53, and definitely will be asked on the USMLE. Here is the second table illustrating the important tumor suppressor genes, and a number of these we've already described when we talked about autosomal dominant diseases that we talked in the genetic disorder section, chapter 6. Remember, VHL stands for von Hippel-Lindau disease, and it's associated with what cancer? Is renal cell carcinoma. Then when we talk about Wilms tumor, the most common pediatric tumor in the kidney, there's two tumor suppressor genes, WT1 and WT2. Then we've already discussed retinoblastoma, and it can be associated with osteogenic sarcoma of the bone. And then P53 is located on the short arm of chromosome 17. It can be associated with a variety of tumors like in the lung, breast, colon, and other parts of the GI tract as examples. Other tumor suppressor genes you can see in familial form of breast cancer. You think of BRCA1 and 2. The function, these are DNA repair proteins that are critical to repair. So if they're defective, then the repair doesn't take place, and then it increases the likelihood of developing breast cancer and or ovarian cancer, especially BRCA1. Then adenomatous polyposis coli, or APC gene, as you remember, it can lead to anywhere between 500 to 3,000 polyps in the colon, and especially the villus type of adenomatous polyp can increase dramatically the likelihood of getting cancer or adenocarcinoma. So that's why when you get the age of 50, in you and I that do not have this autosomal dominant disease, we get a endoscopy of the colon check to see if we have any polyps, and we have to remove them. Obviously, in patients who have thousands of these polyps, they have to get prophylactic colectomies by the time they're age of 30. Otherwise, they're guaranteed to get adenocarcinoma. Then there are colon cancers that are due to DCC, tumor suppressor gene, located on the long arm of chromosome 18. And then we've already described NF1 and NF2 when we talk about neurofibromatosis. And remember, the function of these tumor suppressor genes is to suppress the transduction of cell signals. So they're going to bind to the GTP binding protein, or RAS proto-oncogenes. And they can be associated with neurofibromas, as in the case of NF1, as well as NF2. But remember, NF2 is associated with bilateral acoustic neuromas and meningiomas. So when we talk about the regulation of apoptosis with these tumor suppressor genes, we also have to think of proto-oncogenes as well. When we think of BCL-2, when we talked about apoptosis in Chapter 2 of cell injury, remember BCL-2 prevents apoptosis, and overexpression, especially the most common B-cell neoplasm, is called what? Follicular lymphomas, when you have that translocation of 14-18. And so basically what's on 14 is the genes for the immunoglobulin heavy chains, and on chromosome 18 is the anti-apoptosis gene BCL-2. Genes that are promoting apoptosis, there are a variety. They're called BAX, BAD, BCL-XS, BID, and P53 also, as we remember, promotes apoptosis in mutated cells, and it also stimulates the synthesis of BAX to further augment the apoptosis and preventing mutated cells from entering the cell cycle. c-Myc, as we mentioned, is an important proto-oncogene regulating cellular proliferation, and it interacts with P53 and therefore leads to apoptosis. And when it's associated with BCL-2, it inhibits apoptosis. So the c-Myc has multiple functions because it's a nuclear transgenic regulator, and so it becomes important to realize they have more than one function.